today I'm going to present one of my work entitled Remarkable Intrapopulation Venom Variability in Monocellate Cobra that unveils the neglected aspects of India's snakebite problem. So, this is it. So as we all know, in whole world, there are around 27 million cases of envenoming and 81,000 to 138,000 deaths annually. And in whole world, India is the hotspot for snakebite. And India accounts for half of the global deaths because of snakebite. And in India, we have around 300 snake species and 60 of them are medically important. And because of that, there are around 58,000 fatalities and 140,000 morbidity. Among those 60 medically important snakes, these are the 4 snakes: spectacle cobra, common crate, Russell's viper, and saw-scale viper. These 4 snakes are collectively known as big 4 snakes and considered to be responsible for largest deaths and disability in our country. And these snakes are uniformly distributed all across the Indian subcontinent. And in India, we only have one treatment for snakebite related morbidity and fatalities. That is a polyvalent antivenom, which is raised only for the big 4 snakes. That too for the southern population. So for this. So antivenom is raised for the southern population of these big 4 snakes by injecting this venom to the horses, and the antibody produced by these horses were isolated from their blood, and these antibody we used as an antivenom. Apart from this, we don't have any specific antivenom for another species that cause snakebite apart from these 4 snakes. So, as we discussed, this polyvalent antivenom is only raised for southern population of big 4 snakes. But the venom variability at the intraspecific level across large geographical distances is well documented. And this variability can also influence the effectiveness of snakebite therapy. And as antivenom is manufactured only against one population, that is southern population of big 4 snakes, might not be as much effective as this for the other population of snakes. So, and apart from big 4 snakes, there are other snakes species also, like neglected many snake species, which are distributed to the specific geographical region. And in those regions, bites are more common from these snakes as compared to the big 4 snakes. So the understanding of their venom composition is also limited in our country. So, and we still don't know whether the polyvalent antivenom, which is only raised for the big 4 snakes, can neutralize the bite caused by these snakes or not. So for our study, we are mainly focused on one of the neglected snake species, monocellate cobra. And as we discussed, the intrapopulation venom variation is widely documented for snakes across large geographical distance. But our understanding for the venom variation across finer geographical scale is poorly understood. So for this study, we had selected this monocellate cobra, which is a neglected elapid snake species, and that it is distributed across Indian eastern part, and it is largely uninvestigated, and it is responsible for largest number of death and disability in eastern part of the India. And the venom of this snake is abundant in 3-finger neurotoxin, and we don't have the specific antivenom for this snake. And the polyvalent antivenom, which we have, it is we have for the spectacle cobra, which is even morphologically different, and from the monocellate cobra, it has this V shape. We can see that it has this V shape pattern on the hood, and this monocellate cobra has this circular pattern on its hood. So for this study, we had collected the 25 individual from West Bengal state of India, and these samples are collected from the small geographical region of less than 50-kilometer radius. And these 25 individuals were pooled to the 9 venom samples, and they all are subjected for the crude proteomic analysis by gel electrophoresis. And in this gel image, we can see that in these lower molecular weight toxins, there are the differences in their band pattern and intensity, specifically in these bands, which are mainly the phospholipase A2. We can see the differences in their intensities and in other bands too. Then we did the rigorous analysis by RBHPLC, and we here also we observed the major differences in these fractions. So these fractions are separated on the basis of their affinity towards the solvent, and if we compare this specifically this peak number 3 and 7, the intensity of this peak is very around all of these individuals of same population of same species, and they all are collected from the same geographical region. So if we compare these two samples, sample number 02 and sample number 07, this peak number 3, it has the contrast peak intensity in both of these two samples. So for better understanding, we subjected them to the proteomic analysis through the mass spectrometry. And here also we found that apart from the fact that these two individuals belong to the same population of same species collected from the same geographical region, but still there the relative abundance of some toxin, for example, phospholipase A2 and CRISP, and these are the relative abundance of these toxins vary among these two samples, and the compositional differences can be easily seen in this analysis. Then we evaluated after seeing look into the compositional differences, we had evaluated the variability in enzymatic activity of these toxins, and there also we found that there is a significant differences among the enzymatic activity of these individual of same population of same species. Then we also evaluated the variability in affecting the blood components or blood hemostasis. So when we compare the relative hemolysis through these individuals, we found that some individuals are causing more hemolysis. For example, sample number 04 and 6 as compared to the other individual. And we also found the differences in the affecting the clotting time by extrinsic and intrinsic pathway. Some venom samples are causing are interfering more with the coagulation time than other by both extrinsic and intrinsic pathway. So here also we can clearly see the variability in affecting the pharmacological effects. And after these all in vitro testing, when we evaluated their toxicity, mainly the differences in causing the lethality in the mammal model, we had did the intravenous injection we had intravenously injected these venoms to the mouse, and we found that one of the individual was 3 times more potent as compared to the another one. That is very surprising, because these two individuals belong to the same population of same species, and both are collected from the same geographical region. So after we have clearly observed the interpopulation venom variation in terms of their composition and causing lethality in the mammal model. now we want to know that does this venom variation also affect the antivenom efficacy or not? So for that, we did the in vitro analysis by indirect ELISA. In that, we used the venom as a antigen, and that was coated to the 96 well plate. And then we added the secondary antibody, which is primary antibody, which is mainly the antivenom. And here we tested the recognition potential of four commercially available antivenom: Bharat, Hawkins, Premium, and Serum. These are the polyvalent antivenom which is raised for the big four snakes. And when the obtained binding potential was plotted in terms of optical density. So more the optical density means there is a more binding of antivenom to the venom. And these intensities were plotted against the different dilution of venom, different dilution of antivenom. And here we found that some antivenoms, for example, Premium and Bharat and Hawkins, are showing better binding potential as compared to the this Premium was showing better binding potential as compared to the Hawkins. After evaluating the binding potential of polyvalent antivenom, we had also evaluated the cross-reactivity of Thailand monovalent antivenom. It is an antivenom specifically produced in the Thailand for monocellate cobra, and it is a monovalent antivenom. So surprisingly, we found that the binding potential of this antivenom was more towards the Indian monocellate cobra than the Indian polyvalent antivenom. And when we evaluated the neutralizing potency of these antivenom inside the mouse model, there also we found that these Indian polyvalent antivenom was completely failed to effectively neutralize the monocellate cobra venom, and it is completely failed to reach the marketed claim potency. And the Thailand antivenom, which is given in this light blue color, it is working better for the monocellate cobra as compared to the polyvalent antivenom, but only for one of the individual. So surprisingly, from this outcome, we can say that the outcome of the snakebite therapy mainly depends on the venom of the individual snake responsible for the bite. And that is a problematic scenario. So overall, we had observed the intrapopulation venom variation in the monocellate cobra, and at the finer geographical scale of less than 50 kilometer radius. And we had also observed the significant differences in composition and relative abundance of toxins was observed between the individual of the same population of same species. And we had also observed the stark differences in their biochemical and pharmacological profiles. And even in the potencies was also observed to be different in the mouse model. And this variability can also affect the efficacy of Indian polyvalent antivenom. So the Indian polyvalent antivenom was completely failed to reach the marketed claim potency for the monocellate cobra, and the Thailand and monovalent antivenom was working better as compared to the Indian polyvalent antivenom, but only for the some of the individual. So we can from here we can easily observe that these interpopulation differences in venom can severely affect the effectiveness of snakebite therapy. Thank you.
Thank you very much, Rashmi. Are there any, I see a lot of applauding hands in the chat. Are there any questions from the audience? Feel free to ask directly or write it into the chat. And while you're waiting, I will start with one question. You showed that several of the Najas had different profiles. Could you align them to any biological factor like sex of the animal or maybe the hood marks on the back?
So, gender and all are same for these individual, and all these hood mark is like same for all individual. Those that is mainly specific for the monocled cobra. Yeah.
Okay. Still nothing from the audience. Then I have a second question. You
Okay.
showed that the Thai antivenom seems to have at least some neutralization effect compared to the Indian one. Is a Thai antivenom imported to India? So is it really being used, or did you just show it in the laboratory that it has some potential to be useful?
So we only tested like in the lab, but for the part-time, like for the short-term solution, we can it can be used instead of, because Indian polyvalent antivenom does not have any, like it can't reach the marketed claim potency. For the short-term solution, we can use the Thailand monovalent antivenom. So
Okay.
and it specifically produced for the monocled cobra, and Indian polyvalent antivenom is not produced for the monocled cobra. It is for the spectacle cobra. So it is obvious also that the Thailand antivenom, which is specific for the monocled cobra, it can be better for the for neutralizing their bite.
And one question from the audience queue about that. Going forward, would you plan to look at specimens from different regions in India also?
Sorry, can you repeat?
Did you plan to look on other specimens from different regions?
Other specimen like species or
Specimens, so from the same species, but just different specimens from other regions, since you have now this small
in one.
population.
Okay, so one more, like in another one of our papers from the lab, we have looked into the one more the Naja monocled cobra from Arunachal Pradesh. So there we found that that snake was more cytotoxic, and all of these monocled cobra from West Bengal are more neurotoxic.
And one question more. Were all animals adult, or did they have different
All are
ages?
adult. All are adult.
All adult, okay. How big can the adults be, or how small are the juveniles?
So adult are usually this 3.5 feet to 5 feet monocled cobra. For juvenile, there are like 1.5 feet or something around.